Hello everyone, and welcome to today's webinar, Modeling the Human Brain Using Induced Pluripotent Stem Cells, iPSCs. I'm Alexis Carlson of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Stem Cell Technologies. To learn more, please visit their website at stemcell.com. We encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. I'd like to now introduce our speaker, Dr. Anjana Nityanandam, Director of Operations Stem Cell Lab at St. Jude Children's Hospital. For our full biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Nityanandam, thank you for joining us. You may now begin your presentation.
Hello, everyone. Thank you all for tuning in. Thanks for the introduction. Thanks to LabRoots for inviting me to give this talk and to Stem Cell Technologies for sponsoring it. So as was mentioned in the introduction, I am a neurodevelopmental biologist by training, and I've been working in the IPS field for more than 10 years now. Currently, I am the head of a human stem cell lab, which is a research-based core facility in the developmental neurobiology department at St. Jude. The main goal in our lab is to develop and optimize protocols to generate reliable and robust models of different parts of the human brain. And we work very closely with several PIs in the department. We collaborate with them to develop these models and then to use them to study pediatric neurological diseases. So I've divided my talk into two parts. In the first half, I'll talk about what are induced pluripotent stem cells or iPSCs, some of their applications, how can we make Ips lines, how we characterize them, and one of the key challenges in the field. In the second half, I'll talk about how iPSCs can be used to model the human brain or different parts of the brain, specifically through generating three-dimensional miniature organs called organoids, how these organoids can be characterized, and I'll illustrate a couple of examples from our work in the department to show how organoids can be used for disease modeling and in studying disease biology. Okay, so induced pluripotent stem cells or iPSCs. In 2006, Shinya Yamanaka and Kazutoshi Takahashi at Kyoto University made a stunning discovery. They basically found that if you take fully differentiated somatic cells, fully differentiated fibroblasts from a mouse and introduce four factors, OCT4, SOX2, KLF4, and c-MYC into these cells, then a small fraction of these cells start to undergo a transformation, and within 10 to 12 days, they de-differentiate into these colonies that look remarkably similar to cells that you could isolate and grow from a pre-implantation mouse embryo. They call these cells induced pluripotent stem cells. So they basically are almost exactly like embryonic stem cells. They have all of their properties. This was published in 2006. Very soon, they were able to show that you can do the exact same thing with human skin cells or human fibroblasts and generate human-induced pluripotent stem cells. Now, since then, the technology has advanced a lot. Right now, we have the ability to convert or de-differentiate several different types of somatic cells called donor cells into iPSCs. And this process by which you can convert a somatic cell into a pluripotent stem cell is called reprogramming. So, for example, a peripheral blood mononuclear cells from a blood sample, dental pulp cells from fallen teeth, or epithelial cells from a urine sample, all of these, when we express reprogramming factors listed here, OCT4, SOX2, KLF4, c-MYC, and NANOG and Lin28, when we express these reprogramming factors in these donor cells, a small fraction of them will undergo de-differentiation and reprogram into a pluripotent stem cell. Now, as I said earlier, these are almost exactly like embryonic stem cells, which means that they have the ability to undergo unlimited self-renewal, so they can be expanded indefinitely. and they have the ability to differentiate into cells of all three germ lineages. So they have tri-lineage potential. So they can basically make any cell type in the human body. Now, as you can imagine, that the possibilities are tremendous in terms of what these iPSCs can be used for. The most common application of iPSCs right now is in the field of disease modeling, because these cells now give us the ability to create human disease models. The most simplistic approach to disease modeling using iPSCs would be to take a donor cell, such as fibroblasts, from a healthy individual and from a patient, reprogram them into a control Ips line and a patient-specific Ips line, and then differentiate both these lines in parallel using the same protocol, either two-dimensionally on a substrate composed of extracellular matrix proteins into a two-dimensional neural culture, or they could be differentiated in suspension in 3D to form these self-organizing miniature structures that resemble organs or parts of an organ, and these are called organoids. Although these organoids, of course, are far less complex than an actual organ, but they are a more physiologically relevant model than a two-dimensional monolayer of neural cells. The next step would be then to compare the cellular, molecular, and physiological properties of control versus the patient-specific organoids. And that would basically help you identify disease-specific phenotypes. Now, an alternative approach would be in case of diseases that are complex, where there is a large inter-individual variability, or diseases where the symptoms are wide-ranging. To simplify the study, one can use a control Ips line, and using CRISPR-Cas9 technology, introduce a disease-causing mutation into this control line, and generating an isogenic disease Ips line. So this is the exact same line, has the exact same background as the control line, except for the disease-associated mutation. These two lines, again, can then be differentiated in parallel, either in 2D or in 3D, and then compare the properties of the control and disease-specific organoids to identify disease phenotype. So with this model, one can study, as you can imagine, one can study disease onset, progression mechanism. The models can also be used for high-throughput drug screening, and eventually iPS-derived cells and tissues could be used in cell replacement therapy. Now, later on in the talk, I'll give you a couple of examples of how we in the department have used organoids derived from iPSCs for disease modeling. But right now, I'll illustrate how iPSC-derived neurons are currently being used for drug screening and cell therapy, just to give a couple of examples of how this can be done. So several labs around the world have developed protocols based on what we know from developmental biology studies, protocols to differentiate human iPSCs into spinal motor neurons of the spinal cord. Now, these neurons are significant in a disease called amyotrophic lateral sclerosis, or ALS, which is a progressive a disease where the spinal motor neurons undergo progressive neurodegeneration, which starts with causing muscle weakness, resulting in eventually a complete inability to use your muscles. Now, there is no cure for this disease. So there's a lot of research going on in the ALS field to figure out a way to identify drugs or compounds that could help slow down the degeneration or treat the neurodegenerative condition. Now, all of these labs, they also derived spinal motor neurons from control iPSCs and from ALS patient-specific iPSCs, and looked at the properties of these two neuronal populations. And they were able to identify a bunch of phenotypic features that were quantifiable endpoints to essentially assay the disease phenotype or the severity of the disease. Now, based on this information, a study was done a couple of years back where the authors did a high-throughput drug screening. So they first differentiated an ALS patient-specific Ips line carrying a specific ALS-associated mutation into spinal motor neurons, and then they screened 1,000 different compounds and looked for the basically quantified the endpoints that were assayed previously. to essentially to identify compounds that were able to alleviate the ALS associated phenotype in these patient specific spinal motor neurons. They were able to identify 95 compounds, which were then screened again through motor neurons made from a second patient carrying a different ALS specific mutation. And finally, they narrowed it down to 9 compounds, and then finally one compound, ropinirole, which is a known anti-Parkinson's drug, which is now currently in clinical trials for ALS, for the treatment of ALS. With regard to cell replacement therapy, there are 2 clinical trials currently in progress where the labs have used cells differentiated from human pluripotent stem cells for the purpose of cell therapy. And I want to discuss these 2 examples very briefly here. So in the first case, the first trial is for subacute cervical spinal cord injury, which basically leads to a tetraplegia or quadriplegia, or basically a complete lack of feeling or movement below the neck and shoulder. This trial was started by Geron and continued on by Asterias. So what they did was they took a human pluripotent stem cell and differentiated that into oligodendrocyte precursor cells, which were then transplanted into the cervical spinal cord, the site of injury, right above the site of injury. And in this trial, the data released last year basically shows that this therapy is safe. No adverse effects were reported in any of the participants, and some participants also reported improvement in their motor skills in their upper extremities. The second ongoing trial for cell replacement therapy is from pluripotent stem cells. This is for age-related macular degeneration. This is being done by multiple labs. The condition essentially causes blurred vision, which eventually leads to complete loss of vision. So here what the labs did was differentiate human pluripotent stem cells into retinal pigment epithelial cells. These are the cells that are degenerated in this condition, and transplanted them into the eye. And in this trial, the most recent results show that the therapy is safe, no adverse reactions reported, and some patients reported an improvement in visual acuity. These trials are still ongoing. Now, so ultimately the most promising application of the IPS technology is in the field of personalized medicine. So all of the applications that I just mentioned, they can be used in the context of personalized medicine. What does that mean? It means that we can essentially make iPSCs from any individual, from any given individual, young or old. So you take a patient, either from a skin biopsy or a urine sample or a blood sample, get somatic cells, donor cells, which can then be reprogrammed into a patient-specific iPS line. Now, in this simple schematic by George Daly, it just shows you a couple of examples of how these patient-specific iPS lines can be used for personalized medicine. On one hand, these cells can be differentiated into the cell type that is affected in the patient. And this cell type can then be, then you can basically screen a bunch of therapeutic compounds on these cells and identify drugs that are the most effective for this specific patient and administer those drugs to this patient. On the other hand, using CRISPR technology, the patient-specific iPSCs can be genetically modified to repair the disease-causing mutation that they carry, and then differentiate these repaired cells into a healthy version of the cells that are affected in these patients, and then transplant those cells back into the same patient. So there won't be an immune rejection problem, and essentially the patients are basically getting a modified version of their own cells. So that's just some examples of personalized medicine. Okay, now how can we make iPS lines? The basic workflow involved in generating an iPS line is as follows. So we start with a donor population, let's say fibroblasts, which will be plated on a feeder consisting of inactivated mouse embryonic fibroblasts at a certain cell density. Then there are several ways to introduce the reprogramming factors into these cells that I'll talk about on the next slide. So you introduce the reprogramming factors along with histone deacetylase inhibitors like valproic acid, DNA methyltransferase inhibitors like 5-azacitidine. And the idea behind adding these compounds is to basically ensure an open and accessible chromatin in these donor cells so that they're able to interact with the reprogramming factors. Now, when you do that, depending on the protocol, in 2 to 3 weeks, you will see that a small percentage of the donor fibroblasts have now undergone transformation and differentiation and reprogrammed into a clone. Okay, so these are iPS clones. You may also see, depending on the method used, you may also see several partially transformed cells on the same dish, and those can be ignored. It is important here to note that each iPS clone on the dish comes from a single reprogramming event and a single donor cell. So a single donor cell undergoes reprogramming and gives rise to an iPS clone. Now, this dish can then be live stained for pluripotency markers like CRA160 and SSEA4. And the clones that stain positive, we would then pick and expand these clones. But this time we would expand them on a feeder-free surface comprising of a mix of different extracellular matrix proteins. And this substrate is, the most commonly used substrate is matrigin. Once these clones are expanded, now we know that they express pluripotency markers, but oftentimes the clones may not be able to sustain their pluripotency, so they need to be passaged a few more times to ensure that they're able to sustain the pluripotency. At the end of this process, if you have a good iPS line, it will have a good morphology, tightly packed colonies, no differentiation. A bad iPS line will have tightly packed iPS colonies, but will be surrounded by a lot of spontaneously differentiating cells. And those clones basically can be discarded. We take the good iPS clones and we continue growing them in hypoxic conditions. There have been some studies recently which show that iPS lines grow better or with fewer chromosomal abnormalities, and they're better and healthier when grown in 5% to under hypoxic conditions. And we almost always use MTZR plus from Stem Cell Technologies as a growth media. Now, there are different ways to introduce the reprogramming factors into the donor cells. The three most common methods are: one, using episomal plasmids, which is basically a set of plasmids that code for the reprogramming factors. These can be introduced into the donor cells by nucleofection, for example. They do not integrate into the genome. They stay extrachromosomal where they will express themselves. The second most common method is using Sendai viruses, where basically we've generated a modified non-transmissible form of the Sendai virus vector, which is a single-stranded RNA, which again does not integrate with the genome. It basically replicates and undergoes translation in the cytoplasm. And the third method, which is my personal favorite, is using synthetic modified mRNAs, which code for the reprogramming factors. These mRNAs are modified, as shown here, to basically not trigger the cell's the innate cellular immune response, which can affect the stability of the mRNA and translation efficiency. So the modification basically makes sure that the mRNA is stable and is able to undergo translation efficiently. And here's a table summarizing some of the pros and cons of each of these methods. Briefly, episomal plasmids are affordable, but they do require a large number of iPS clones to be screened, because the rate of aneuploidy in the iPS lines that you get from using episomal plasmids is a bit higher than the other methods. Plus, one needs to passage iPS lines at least 10 times before you can completely get rid of the plasmids from the cytoplasm of those iPSCs. And that is important because we don't want these reprogramming factors to continue expressing ectopically when you're trying to differentiate your iPS lines towards specific cell lineages. Sendai viruses are very commonly used. They are expensive, but they reprogram very efficiently. They are highly reliable. The aneuploidy rate is low, but once again, you need to do about 15 passages to completely get rid of those viral vectors from the iPS lines before downstream differentiation. The mRNA methods are, the mRNA method is expensive, but offers the highest efficiency of reprogramming, very low aneuploidy rate, and one needs to do only about 5 passages before the lines are ready for downstream differentiation. Okay, so once you have picked your iPS clones and expanded them, made sure that they are able to sustain their pluripotency, the next step is to characterize them. Next step is to validate them and make sure that they are truly pluripotent. There are 3 main aspects of the characterization. The first one is to confirm the expression of pluripotency genes. which can be done by simple immunostaining. There are a bunch of markers, cell surface markers, as well as transcription factors that are expressed by pluripotent cells. To do a more quantitative assay, one can do a flow cytometric analysis. As shown here are two Ips lines. The first one, almost all cells express both pluripotency markers at very high levels. In the second line, you can see there is a small fraction of cells in this population that do not express either of the pluripotency markers, and that's a sign of spontaneous differentiation in this line. So it's not as good a line as the first one. For an even deeper analysis, you could analyze your cells on a microfluidic array loaded with Stackman probes, such as this one, which will basically look at the expression of several genes associated with pluripotency and differentiation. And one could do a similar assay on a gene expression microarray, such as this one from Affymetrix. The second aspect of characterization is to look at the trilineage potential of your Ips line, whether it is able to differentiate into all cells of all three germ lineages, ectoderm, mesoderm, and endoderm. Now, there are two ways to do this. You can do it through an unbiased differentiation approach, where you basically let the iPSCs undergo default differentiation into a culture which is a mix of cells from different lineages without pushing the line towards any specific lineage. An alternative approach is to do a directed differentiation, where you're essentially pushing your Ips line specifically towards ectodermal, mesodermal, and endodermal lineages by adding a specific signaling molecules and morphogens into the medium. This is the method that we prefer to use in our lab. There are also several commercially available kits, such as this one from Stem Cell Technologies, which allows you to do your trilineage differentiation through a directed approach. Okay, so once you have your culture, these can be analyzed by RT-qPCR for markers of the different lineages. So in our lab, we use a customized Stackman array where we're basically looking at the expression of 7 genes for each lineage. And but you can also do a deeper analysis, for example, using a scorecard assay, such as this one, which basically looks at the expression of a lot more markers for each of the lineages. Okay, so and finally, the third aspect of characterization of Ips lines is looking at genomic integrity. Now, Ips lines are known to carry structural abnormalities and copy number variations. Some of those are listed here. The most commonly occurring abnormalities are listed here, and most of them confer a growth advantage to the cells. So the abnormal cells will quickly take over the entire culture. So it's very important to be able to identify them early on. Now, what is the reason for these structural variations in Ips lines? There are several causes. For example, if your donor population is carrying a specific cohort of somatic mutations, and that basically gets carried on to the Ips line that was generated from reprogramming that particular donor cell. Another reason could be a suboptimal culture condition. So we know that the longer you culture Ips lines in vitro, the more the chances of them acquiring genomic problems as well as epigenetic mutations. In addition to that, for example, single cell passaging versus passaging your lines as pieces of colonies makes a difference in terms of the genomic health of these lines. If you let your cultures grow beyond 80% confluency, that seems to affect their karyotype as well. And as I said, prolonged in vitro culture. So basically, optimal handling of these Ips lines, not letting them overgrow, always passaging them in pieces and not as single cells, is important to avoid these structural abnormalities and copy number variations. So how do we make sure that the lines are good genomically? The simplest assays are G banding and spectral karyotyping. So here, for example, we have a line where 93% of the population has a normal karyotype, but 7% of these cells have a trisomy in chromosome 20. And here we have a line where there is a massive duplicate. We have a normal line, and then there is a line where we have a massive duplication in chromosome 20. So chromosome 20 abnormalities are very common in human ES and Ips lines. For a deeper analysis, you can also analyze these lines for a higher resolution analysis. You can also look at them on SNP arrays, such as this one from Illumina. The array will basically detect copy number variations, indels, translocations, and SNPs as long as they're present in at least 20% of the population. So it gives you a digital karyotype of the line. And several labs also prefer to do whole genome sequencing. So you can detect all your deletions, insertions, single nuclear variants, de novo variants, etc. Okay, so one of the key challenges in the IPS field is a phenomena called interline variability. It basically means that if you take a population of donor cells and you derive two IPS lines, in the same reprogramming experiment, you derive two independent IPS clones or IPS lines from this donor population and differentiate both of these lines using the same protocol, the lines might actually behave slightly different from each other in terms of their differentiation potential and lineage bias. So now, why is that? Although they were both derived from the same population of donor cells in the same reprogramming experiment. So there could be many causes for interline variability. The two main causes that I'll talk about here. One is if you have a heterogeneous donor population. So as I mentioned earlier, every IPS clone or IPS line comes from the reprogramming of a single donor cell as a result of a single reprogramming event. So if you have a heterogeneous population of donor cells, where the different cells carry different cohorts of somatic mutations, those mutations basically are going to get transferred over to the IPS lines that come from these cells, resulting in a heterogeneous group of IPS lines derived from the same donor population. The second reason could be aberrant epigenetic resetting. So what is epigenetic resetting? Now, during reprogramming, you start with a donor somatic cell where this is a fully differentiated cell. So all the pluripotency genes are repressed, which means they carry hypermethylated promoters and repressive histone modifications such as the lysine 9 and lysine 27 trimethylation. Whereas all the cell type specific genes are active, so they have hypomethylated promoters and they carry activating histone modifications like lysine 4 methylation and acetylation. Now, during reprogramming, this somatic cell erases all of its epigenetic marks and reestablishes new epigenetic marks. So basically, it reestablishes its epigenetic landscape so that the resulting IPS line will have all pluripotency genes active with hypomethylated promoters and activating histone modifications, and all the cell type specific genes in the IPS line will be repressed. So hypermethylated promoters and repressive histone modifications. Now, if this process of this process is called epigenetic resetting, where the somatic cell is basically erasing all of its epigenetic marks and re-establishing new marks to become a pluripotent stem cell. Now, if this process of epigenetic resetting happens perfectly, then when you differentiate this IPS line, it should be able to differentiate into cells of all three lineages with equal efficiency and no bias. Oftentimes, however, this does not happen perfectly, and the resetting process can often be incomplete or aberrant. Now, this is a random process, and it can vary between every single reprogramming event. But what happens is that you have a donor somatic cell which undergoes reprogramming into an IPS line, which does express the pluripotency genes. However, the repression of the cell type specific genes might be incomplete or aberrant. As a result, you have an IPS line which is pluripotent, so it is able to differentiate into cells of all three lineages. However, because of this incomplete repression of cell type specific genes, it might have a skewed differentiation potential or a lineage bias. Now, if you derive, let's say, 6 different IPS clones from a single donor population, now because epigenetic resetting aberrant epigenetic resetting is random, and because it can be different in every single reprogramming event, and because all of these 6 clones, each clone comes from a single reprogramming event, you end up with 6 clones which might have slightly different epigenetic profiles and therefore slightly different lineage bias, resulting in interline variability. Now, eventually, we might be able to optimize and standardize a method of reprogramming that gives us the highest quality IPS lines with complete epigenetic resetting and no lineage bias. However, right now, the most common way to address this variability issue is by screening. So the idea is to generate multiple clones from each donor. So let's say 6 IPS clones from each donor, and then you would screen these clones and identify at least 3 clones per donor, which are best suited for your purposes, and then differentiate all 3 iPS clones from each donor into organoids or whatever downstream differentiation that you want to do. And then you would analyze basically organoids from all of those 3 clones. So the idea here is that if you see a phenotype in organoids from all 3 iPS clones, then you can be confident that that phenotype is a consequence of the genotype of the individual and not because of some inherent bias in the line itself. So this screening of iPS lines, there are several ways to do that. I'll talk about 2 assays here. The assay that we do most commonly or routinely in my lab is a cellular assay, because our goal is neurological disease modeling, or therefore neural differentiation. We do a rosette formation assay where we take iPS lines and we aggregate them in these agrivals to form embryoid bodies in this neural induction media from stem cell technologies. After a few days, we then plate these embryoid bodies on a substrate because they are growing in this neural induction media. They should be, for a good iPS line, the embryoid bodies should be able to form these neuroepithelial structures called rosettes. If you have a line with high neurogenic potential, 95 to 100%, it will be 95 to 100% efficient in forming neural rosettes. But if you have a bad iPS line, then either the efficiency will be low, or you'll have a culture which would be a mix of neural rosettes and non-neural cells. So a cellular assay such as this one, especially for neural differentiation, is quick and simple to identify clones which have a decent neurogenic potential, or which have a good neurogenic potential. For a deeper analysis, you can also analyze your iPS lines on methylation arrays, such as this one from Illumina. And you could either look at the global methylation profile of your iPS lines and compare them to the donor population, and that might help you predict lineage bias. Or you can look at the methylation at specific promoters of genes associated with pluripotency and differentiation, and this sort of methylation pattern can again help you predict lineage bias. So, for example, in an ideal situation, if you have a high quality iPS line which does not have lineage bias, then the promoters of all differentiation of cell type specific genes of ectodermal, mesodermal, and endodermal lineages should all have hypermethylated promoters. Okay, so to summarize the first part of my talk, I basically showed you that iPSCs can be derived from multiple donor cell types using multiple non-integrating approaches. iPSCs can undergo unlimited self-renewal and have trilineage potential. To characterize and validate iPS lines, we need to look at the expression of pluripotency markers. We need to check their genomic integrity periodically, as well as demonstrate their ability to differentiate into ectoderm, endoderm, and mesodermal lineages. The applications of iPS technology in disease modeling, drug screening, cell replacement therapy, and most importantly, this allows for personalized medicine, which is still a few years away, but that is the eventual goal. Interline variability amongst iPS lines derived from the same donor is a key challenge. At the moment, the most common way to address this issue is by deriving and screening multiple iPS clones per donor. And to do the screening, you can do cellular assays or methylation arrays. Okay, so now the second part of my talk, I'll be talking about how we can use iPSCs to model the human brain. Now, differentiating iPSCs into brain organoids broadly comprises of 4 main phases. So these 4 phases are neural induction, expansion, differentiation, and maturation. So neural induction begins with dissociating iPSCs into single cells and reaggregating them at a defined number of with a defined number of cells per aggregate in a low attachment plate, such as a low attachment 96 well plate. These cells will reaggregate in the right media to form these embryoid bodies. The embryoid bodies, basically the cells of the embryoid bodies, depending on the growth factors and cytokines and inhibitors provided in the media, they'll undergo regionalization and cell fate specification, and they will proliferate into these neuroepithelial, these self-organizing neuroepithelial structures that have a lumen and they are surrounded by region-specific progenitors, and they have an apical basal polarity. The next phase is neural expansion, where these region-specific progenitors start proliferating, they expand. They also get specified into lineage-specific precursors, and some of these precursors also start differentiating. For example, in this double reporter line, you can see that the organoid still is comprised mainly of progenitors in green, but some of these progenitors are starting to differentiate, shown in red here. So the onset of differentiation starts with the neural expansion phase. The differentiation of the lineage-specific precursors continues in the neuronal differentiation phase. During this phase, it is important to provide conditions for good nutrient and gas exchange for these organoids. And so we typically grow these organoids in these mini bioreactors, which basically will agitate the organoids and allow for nutrient exchange. And we know from comparing RNA-seq data of retinal organoids grown with and without agitation that agitation significantly reduces hypoxia-induced pathways in these organoids. And organoids just grow better when they move around like this, as opposed to when they are just grown immobile in a plate. So that's neural differentiation, and this is then followed by maturation, for which we almost extensively use the brain fizz media, again from stem cell technologies. During the maturation phase, as the organoids mature, you'll start seeing the expression of synaptic proteins, expression of, for example, here glutamate transporter. And for organoids such as cortical organoids, when you plate these organoids on multi-electrode arrays and look at the spontaneous activity, you'll start seeing spontaneous neuronal networks, network bursting and oscillations. All signs of the fact that the neurons within these organoids are maturing. So those are broadly the four main phases. Now, different labs have their own customized, optimized protocol for differentiating iPSCs into different types of organoids of the brain. However, there are also commercially available kits, such as this one from Stem Cell Technologies, which can also be used for differentiating iPSCs into brain organoids. Okay, so now once you have an organoid, the next step is to determine how closely does this organoid resemble the region of the brain that you're trying to model or mimic. Now, characterizing these models, again, involves three main aspects. There are several assays that you can do for characterizing your models, but the three main aspects of it. The first one is immunostaining, which involves simply sectioning the organoids and staining them staining them using antibodies against specific cell type specific markers and genes. Here, for example, we have a bunch of rostral hindbrain organoids that we made in the lab, and we've sectioned and stained them in collaboration with the Northcott lab to look for the presence of, to check the presence of a certain specific cell type. So immunostaining is the advantage of a quick immunostaining is, first of all, it's a good way to determine if you are going in the right direction, if you are differentiating the right lineages, just to confirm the presence of specific cell types that you expect to see in the brain region that you're trying to model. It also shows you cellular organization and cytoarchitecture within the organoid itself. If the cells are layered, they're laminated, and the distribution and localization of the different cell types relative to each other. So it gives you an idea of how well the organoids, the cells are organized within the organoid structure. And is it similar to what you would see in vivo? The second aspect of characterization is looking at the transcription profile. So you could do a bunch of RT-qPCRs, or you can do bulk RNA-seq, or more commonly, or what's becoming more and more common in the field, is to do single-cell RNA-seq. And here are some examples of single-cell RNA-seq data published by different labs. The advantage is that this basically, the assay tells you what proportion of different cell types are present in your organoid. If you have and it also tells you if you have cell types that you do not want in your organoid system. So, so cells from the neighboring lineages or neighboring parts of the brain, the undesirable cell types. So, so this is a more quantitative analysis of your organoid, of the composition of your organoid. And the third aspect of characterization is to perform a functional assay. Because we have brain organoids, so we are the functional assay is essentially the best way to sort of demonstrate the physiological relevance of your organoid model. So for brain organoids, we want to know if they have a functional neurons, if they have neurons that can actually form a neuronal network and function and fire spontaneously. So in our, again, there are different ways to do this, but in our lab, we typically plate organoids on these multi-electrode arrays. We have a system from Axion Biosystems. Here we have a grid of 16 electrodes, and there's a cortical organoid that we have plated on these. The multi-electrode array essentially measures extracellular field potential, which is a direct consequence of the firing of action potential by the neurons, which are in contact with these electrodes. Now, when you have in cortical organoids, when you have a mature cortical organoid, these neurons with a dense network of neurons, they will basically communicate with each other and fire synchronously, as shown here. Where you can, so basically each of these raster plots is from a separate organoid, and on the x-axis is the time, and on y-axis is the 16 different electrodes. Basically, each vertical line here represents the firing of an action potential. So as you can see here, you have a bunch of electrodes firing synchronously, or firing at the same time, which basically suggests that there is a network of neurons across these 16 electrodes, and this is what you would expect in 100-day-old cortical organoids. Notice here that each of these organoids has a slightly different pattern of firing, but they all are able to fire these neuronal, these network bursting or network oscillations. Okay, so a challenge, just like Ips lines, the challenge in the organoid field is variability amongst organoids. Now, organoids generated within the same experiment, within the same batch, as well as organoids between independent experiments using the exact same protocol, can be quite variable. We have seen variability in yield. We've seen variability in size. For example, here we have a bunch of cortical organoids, which are highly variable in size. We've seen variability in the morphology of the organoids. So here we have a batch which has a bunch of good-looking, dense organoids with smooth, translucent edges, but we also have a ton of cystic organoids. Here is a reporter line, a bunch of cortical organoids expressing a reporter. Now we have some organoids expressing the reporter very efficiently and strongly, and we have a bunch of organoids with no reporter expression. This is within the same batch. And here we have a bunch of caudal forebrain organoids expressing a different reporter. Again, you can see there are some organoids with strong, full expression of reporter, some with weak expression, some without any expression at all, and some expressing the reporter in patches throughout the organoid. So the variability can be quite significant. So what is the reason for this variability? There could be many reasons, but the two reasons that we are focusing on resolving in the lab are first, if you start with a heterogeneous population of iPSCs. So if you have a population of iPS cells which are already heterogeneous with regard to their genetic, with regard to their genomic abnormalities or their epigenetic profile, because the process of differentiation starts with dissociating these iPSCs into single cells and then reaggregating them in defined cell numbers. The proportion of good and bad iPS cells that you end up plating in every single well of that 96 well plate might be different. So your starting EB might be actually very heterogeneous within a single 96 well plate, as a result of which the organoids that you get eventually from those EBs will also be variable. So a heterogeneous starting population of iPSCs could lead to huge variability. The second reason could be low efficiency of the differentiation protocol. So whatever morphogens, whatever signaling molecules and cytokines and inhibitors you're adding to the media, if the efficiency of them influencing causing cell phase specification, influencing differentiation is not complete, then that can also lead to a lot of variability depending on how many cells or which cells are getting exposed to the morphogens and which cells are able to actually respond to the morphogens. So right now, to address the variability issues, to address the heterogeneity issue, what we do is frequent karyotyping. So the general consensus in the field is to karyotype your iPS lines every 5 passages, but we probably do it more frequently than that. So frequent karyotyping just by G-banding and spectral karyotyping will give you an idea of how, if your lines are still maintaining their genomic health. However, we don't know about the epigenetic changes taking place in these lines. And then optimal handling, like as I said before, handling of the IPS lines, never letting them overgrow or grow beyond 80% confluency, for example, and just passaging them and maintaining them, the cryopreservation, just optimal handling conditions of the IPS lines also helps in maintaining maintaining a healthy line and therefore reducing variability downstream. And in order to address the efficiency issue of the differentiation protocol that you're using, my recommendation would be to use a reporter line wherever possible. Now, we've had issues with some of the protocols that we've tried to develop to model different parts of the brain, and using a reporter line is extremely helpful in optimizing your protocol, in determining the variability in your protocol, the variability in your system, in determining the efficiency of your system. So wherever possible, use a reporter line. And then we can also use these reporter lines to essentially screen organoids at an early stage, screen organoids that are expressing the reporters versus organoids that don't express the reporter well, for example. And before doing the more detailed experiments or downstream analysis, at an early point during differentiation, you could use a reporter line to screen the healthy organoids or the good organoids. So I'll give you an example here. So here we have a culture, a batch of caudal forebrain organoids expressing a caudal forebrain specific reporter. Now, you can see this organoid is a mix of, this population is a mix of organoids expressing the reporter, marked here with the yellow asterisk. And then we also have organoids marked here with the blue asterisks, which don't express the reporter at all. And we have all of these organoids growing in a single batch. So now what we did was we basically compared the gene expression profile by bulk RNA-seq of organoids, which were made from a non-reporter line. So we basically collected a bunch of unscreened organoids. And then we made organoids using the same protocol from the reporter line, and we screened for the reporter expressing organoids and then did a bulk RNA-seq on those organoids. Now, in this heat map, which is a subset of the RNA-seq data, we are looking at the expression of caudal forebrain genes and dorsal forebrain genes, and each row here represents a single organoid, one organoid, basically. And as you can see in the reporter screen, if you look at the reporter screen organoids, they almost all look almost exactly similar, with a high expression of caudal forebrain genes and low expression of dorsal forebrain genes. But if you look at the unscreened organoids, not only do they have a completely different expression profile, they also the organoids also differ from each other quite a bit in terms of the level of expression of the different genes. So if wherever possible, use a reporter line and use that to screen organoids before you start doing the more the bigger downstream experiments. Okay, so now I want to give you two examples of disease modeling using iPSCs and iPSC-derived organoids that we have done in the lab in collaboration with PIs in the developmental neurobiology department at St. Jude. The first example is a project we did in collaboration with Dr. Mike Dyer, where the goal was to model human retinoblastoma in order to identify the cell of origin of this tumor and to provide a model for preclinical testing. Now, our role or our job in this collaboration was to develop and optimize a highly reliable and efficient protocol that could differentiate any IPS line into a retinal organoid, and which is basically what we worked on. We used a couple of reporter lines here, which the Dyer lab received as gifts from their collaborators. And using these reporter lines, we basically optimized a protocol whereby we could differentiate iPSCs from 15 different individuals efficiently into retinal organoids with a good yield and a decent morphology. We also saw that our retinal organoids, they are functional. So we've seen retinal pigment epithelial cells juxtaposed with photoreceptors, which basically suggests the formation of a functional photoreceptor signaling. And we've also seen that older retinal organoids, when we plate them on a multi-electrode array and provide them light stimulus, we were able to record a wavelength-specific light response from the retinal ganglion cells in these organoids, which basically suggests a functional pathway between photoreceptors and retinal ganglion cells. So we have functional retinal organoids that we could derive from multiple individuals. So once we had the protocol, we worked very closely with Jackie, who's an excellent postdoc in the Dyer lab. So what Jackie basically did was, Jackie and her other collaborators, they basically collected donor cells from 15 different retinoblastoma patients, carrying different, a wide-ranging mutations in the RB1 locus. They then generated Ips lines from these 15 different individuals. We then screened the Ips clones, and we selected the clones. We did a neural rosette formation assay, and we selected the clones that had high neurogenic potential. And we were then able to differentiate Ips lines from each of the 15 patients into retinal organoids. Jackie then took these organoids, and she at 45 days of differentiation, dissociated them into single cells and injected them intravitreally into immunocompromised mice. And after about a year, she was able to see that 7 out of the 15 organoids, 15 patient-specific organoids, were able to generate a retinoblastoma, which is a tumor of the eye, in these mice. And we could, they could basically confirm this by histology. And they also looked at the DNA methylation profile in these organoid-derived retinoblastomas, and they found that the organoid tumors, they clustered with the patient's tumors and the xenografts. They also did single-cell RNA-seq on these retinoblastomas and found that the organoid-derived tumors, again, they look remarkably similar to the patient-derived tumors as well as the patient-derived xenografts with regard to cell type identity and proliferation. And this work we are about to publish this work. It's in the final stages of submission. In the second example, we worked in collaboration with Dr. Fabio De Montes. The De Montes lab studies aging and neurodegeneration in Drosophila as their model system. But what they wanted to know was, they wanted to see if stress-induced neurodegeneration is something that can be modeled in a human organoid system. So the De Montes lab, basically working with Drosophila, they identified a unique pathway. They found that if you apply proteasomal stress to skeletal muscle in Drosophila, the muscle releases a myokine called amiral, which is able to induce a neuroprotective effect in the retina and brain of the Drosophila. So, for example, they found that Drosophila that express a pathogenic form of the Huntington protein, they have these protein aggregates in the eye. However, if you overexpress amiral in the skeletal muscle of these Drosophila, these aggregates disappear. Similarly, Drosophila that express a mutant form of the tau protein, they have degenerated photoreceptors because of this protein aggregation. However, if you overexpress amiral in the skeletal muscles, then this degeneration again can be rectified. So they looked into this pathway a little bit more, and they found that this neuroprotective effect of amiral is mediated via maltose. And so the De Montes lab wanted to know if this pathway, which is active in Drosophila, is it conserved in humans? Is this something that can be seen in the human brain, as in a human system as well? So for that we generated cortical organoids. And we decided to test this pathway in cortical organoids by looking at heat-induced neurodegeneration. Now, the lab did a bunch of assays, but there's one assay that I'm going to highlight here, which we did in my lab. So we basically took mature cortical organoids. These are 2 organoids. One treated control, one is a control, and the other was treated with maltose, which the lab showed in Drosophila has this neuroprotective effect. And then we used heat shock as a means to induce neurodegeneration. Now, before heat shock, both of these organoids have strong spontaneous network activity. After 7 hours of heat shock, maltose-treated organoids continued to have strong activity, but the activity completely disappeared in the control organoids because of the degeneration induced by the heat shock. And even after 18 hours of recovery following the heat shock, following the end of the heat shock, the maltose-treated organoids still have strong spontaneous network activity, whereas the control organoids barely recovered any activity at all. So this, we recently published this work in Cell Metabolism. So this basically, this is a good example of how human brain organoids can be used to validate findings from a different model system. In this case, Drosophila, which I think is pretty cool. So to summarize this part of my talk, I basically showed you that iPSCs can be differentiated into these self-organizing 3D structures that mimic parts of the brain called organoids. And these can be, depending on the protocol that you design and develop, these can be region specific. However, variability amongst organoids is a problem, and using a reporter line and screening organoids for specific endpoints or for specific phenotypic features is very useful to address this variability. To characterize brain organoids, the different assays can be done, but the three sort of fundamental aspects of characterization involve immunostaining to look at the cytoarchitecture within the organoid, transcriptome analysis to look at the proportion of different cell types that are present within the organoid and how pure the organoid is, whether it's contaminated by lineages from neighboring regions of the brain, and by doing a functional assay, which essentially is to show its physiological relevance. I showed you that we in our department, we've used human iPSCs to generate patient-specific retinoblastoma, patient-specific retinal organoids, which were able to generate a patient-specific retinoblastoma in immunocompromised mice. And these organoid-derived retinoblastomas have the same molecular profile as patient tumors. I also showed you that we've also, in our department, have been able to use iPSCs to model stress-induced neurodegeneration by looking at the neuronal activity, spontaneous neuronal network activity in mature cortical organoids in response to thermal stress and in response to treatment with and without neuroprotective factors that were discovered in the lab. So basically, in this project, we were able to translate findings from Drosophila to humans, which I think is pretty exciting. And finally, I want to thank Kyle and Priyanka, who are members of my lab, who are very hardworking, excellent, brilliant people that I was very fortunate to hire. I want to thank all the members of my department that we work very closely with, that we collaborate with, members of the Dyer Lab, Zakharenko, Northcott, and Demontis Labs. The DNV operations support team, the cytogenetics core at St. Jude does all our karyotyping, and the Center for Advanced Genomic Engineering, which is run by Dr. Chandra Miller. They have basically created a lot of reporter lines as well as isogenic disease specific lines for us for multiple projects that we did. And finally, St. Jude is an amazing place to work at, and if anyone's interested looking for a job, I would highly, highly recommend coming to St. Jude. It's just an amazing place to work at. Thank you everyone for listening, and I will be very happy to take questions.
Thank you, Dr. Nityanandam, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. So let's get started. We've already got some great questions that came in from the audience. So we're going to start with this one first. How much optimization time do you typically plan for when bringing in a new line?
So, it kind of depends on what we want to do downstream with the line. If it is a long-term differentiation, if it is a quick assay, then maybe just a couple of months. If it is a long-term differentiation, then we kind of plan to do several experiments in parallel. It depends on the protocol that we are using, so it's kind of hard to predict. If we're using a protocol that we've done many times, that has been fully optimized, then it's easier to tweak it, play around with certain conditions just to make it work better for the line. But if we have a protocol that we're still trying to figure out, we're still working through, then that obviously takes longer. So it's kind of hard to determine. But so anytime we start working with a new line, the first thing we do is karyotype them That's very, very important, because if your line has a bad karyotype, or even if you have like 2% of the population with a chromosomal abnormality, that's going to quickly take over the entire culture, and that's going to ruin all your downstream experiments. So karyotyping is the first thing that we do. And then we'll do like a neural rosette formation assay to make sure that it is able to generate neural cells at a decent efficiency. And then everything after that is it just depends on the protocol and the aim of the experiment.
Great, thank you so much. Moving on to our next question, which is a two-part question for you. So what is the best way to keep organoids healthy in long-term culture? And the second part is, are bioreactors the only option?
Yeah, that's a good question. So for long-term culture, we've tried different ways to maintain organoids long-term. And it's especially important if you want to get to a stage where the neurons are mature and they are firing, and if you want to like study all of that, then you need to keep the organoids healthy and maturing and differentiating for a long time. So the thing that works best for us, one is, as I mentioned in my presentation, agitation. So bioreactors are expensive. If you do not want to purchase a bioreactor, I'm hoping that bioreactors become more and more common in labs, and the price goes down, and there are like multiple. Right now, we don't really have that many commercially available systems that are affordable and can be used, established easily in the labs. The alternative would be to use orbital shakers, which we also use. So you can essentially put your organoid in a 24-well plate, low attachment plate, or a 48-well low attachment plate, and we place them on an orbital shaker. And then we move them at like 80 RPM or 90 RPM. And the entire orbital shaker is basically inside the incubator. And there are several that you can purchase, which are small, compact, and they don't cause any vibration. You can just fit them inside the incubator and move your organoids around. That also helps a lot with the growth of the organoids. The longer the organoids grow in culture, the more necrosis they will start to get in the center. So all organoids will have a necrotic core. They'll have a necrotic center because the nutrients and gas, they just cannot reach the center of the organoid. That is unavoidable. So one thing you can do to reduce the size of necrosis is cut the organoids into smaller pieces periodically. So after a certain age, let's say after like day 35 or 40, every 1 or 2 weeks, we'll take the organoids and we'll cut them into smaller pieces, usually in half or in like 3 pieces, and then put them back in the bioreactor and continue. So cutting the organoids into pieces every now and then, that really helps with controlling the size of the necrosis in the center of the organoid and keeping the organoid growing long term. And the last thing I would say is changing media frequently. I mean, I know media is expensive and we don't want to waste media, but we've noticed that if you change the entire, instead of changing half the media, if you change the entire media in the culture and do it every, let's say, 4 or 5 days, that's very, very helpful in keeping the culture healthy.
Thank you so much. And it looks like we have time for one more question. So this audience member was wondering if you could speak a little bit more to the variability in PSC line in terms of working with them, for example, like handling with them.
So the iPSC lines themselves, when you start working with different iPSC lines, you notice that they can have the undifferentiated pure IPS line itself can have like different morphologies. So there is no, just by looking at an IPS line, it's kind of, sometimes it can be hard to tell whether it's going to be a good line or not, or if it's going to, if it's going to differ from the other IPS lines when you start differentiating it into neurons, because the morphology of the IPS lines kind of varies a bit sometimes. We haven't really found any correlation between what the IPS line looks like under the microscope versus, with what type of neurons it's able to differentiate into. The only thing I can suggest is that, as I said, making sure that the IPS lines don't become overconfluent, that is very, very important. And if we do have IPS lines that are variable in terms of like just the morphology or the appearance of the lines, oftentimes, if it's a precious sample, then we'll do a FACS purification. So we'll basically just stain the lines with 2 cell surface pluripotency markers, and we'll do like a FACS purification and purify just the cells that are that have high expression of the pluripotency markers and grow those and discard everything else. That is only for like very precious samples because that's not a foolproof method. A lot of those cells purified by FACS will also, after a couple of passages, they might start differentiating as well. So, but yeah, so in terms of handling IPS lines, there's not much that you can do to like avoid variability except karyotyping them frequently, making sure they're not overgrown, just handling them properly, and the variability, the majority of the variability comes from the reprogramming process, the aberrant epigenetic resetting, the reprogramming process itself. So instead of, in fact, my personal recommendation would be, instead of spending time trying to like, make a culture better, I would say just derive multiple clones from the donor, because it's very easy to derive multiple clones from the donor. It's not really that much more work than deriving 3 clones. And then just screen the clones before expanding and banking them. Just screen the clones and identify the ones that seem best for your purpose, and then expand and bank those.
Thank you so much, Dr. Nityanandam, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Stem Cell Technologies, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for all of their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us. Until next time, bye-bye, everyone.